This tutorial will give you an overview of the asthma-map, showing the different levels of resolution and how to navigate the submaps. Later videos will show how to use some of the powerful features of this platform, but for now we'll take an overview of the map as a whole. The asthma-map is composed of 3 different layers. Each of these layers describes the same molecular pathways, but with a different level of granularity and with a different purpose in mind. The 3 layers are cellular interactions. This is a high-level view showing the cell types involved in asthma and how they communicate with each other. This level provides a visual overview and links to the more detailed submaps. We will explore it in more detail in a moment. The second layer is molecular relations. This consists of a collection of submaps describing the molecular interactions in each cell type. Finally, the biochemical mechanisms layer, the most fine-grained level, contains details about the biochemistry behind the most important pathways in asthma. The map can be explored using the Minerva platform, and the remainder of this tutorial will show how to navigate the asthma map using the web-based Minerva tool. Following the link, we'll open the overview map in the Minerva browser. Using this browser, we can navigate through the different layers and submaps in the asthma map project. We will now go through the 3 layers one by one. The cellular interactions map provides an overview of the different cell types involved in asthma. The interactions between cells are presented together with the molecular species mediating these interactions. Clicking on any of these proteins brings up detailed information in the sidebar. It is also possible to search for a molecule. To do this, type its name in the search field and hit enter. The search results indicate all instances of this protein in all of the submaps. Each one of these instances is linked to the submap where it is found. The submaps can also be accessed using the links in the main map, or from the List in the Submaps tab. Most of these submaps are part of the molecular relations layer. Selecting a submap by clicking on the magnifying glass icon will open it in its own window. The result of our active search is highlighted, and details are visible in the Search tab. You can clear this if you like by clicking the Global Clear button. Zooming out shows the scope of this submap and how it represents a functional interaction map of the core molecular pathways in this cell type relevant to asthma. In fact, this structured representation can be readily adapted for logical modeling. Look at the asthma-map website for more details on this aspect of the project. There are currently 2 submaps in the biochemical mechanisms layer describing the mast cell and the eicosanoid pathway. As can be seen when loading one of these submaps, they contain very detailed information at the level of biochemical reactions. These submaps offer scope for powerful interactive analyses, which are introduced in the accompanying tutorials. The ecosystem of the multi-layer asthma map enables researchers and clinicians to not only gain an overview of the cellular and molecular processes involved in asthma, but to dig much deeper into the functional and biochemical mechanisms underlying the disease. Navigating the map in Minerva provides an interactive experience with powerful tools on hand, and the data can also all be downloaded in a range of formats for offline use. We hope you'll enjoy exploring the map yourself at https://asthma-map.org.